Good morning. Can you hear me? Good. I'm really pleased to be here, to be able to interact with you as well as with this short presentation of 15 minutes of fame. It's a big topic, so I decided to take the bullet train approach, and we'll be seeing a lot of things that hopefully will stimulate conversation as the morning progresses. So without further ado, I just want to start with why precision assessment. Well, it's already been alluded to in the opening remarks that conventional assessments, certainly clinical assessments, are, I'll just say, inadequate. They are brief, episodic. They're often relying on subjective information. They're clinic-based, often not convenient either for the participants or the clinicians. And I think the most important thing is they're not ecologically bound. They don't necessarily really represent what's happening every day in the real world, as these photos on the bottom from patients of mine show. And we know that the data that's collected is, as again, this is actually a slide already presented, it shows the immense heterogeneity. On the lower left are two figures from AGNI data, showing the data as COG13 results of people with great individual as well as tri-individual heterogeneity over time, as well as on the right is from a longitudinal study of Korea, South Korea, just showing the CDR sum of the boxes results with subjective cognitive impairment, MS, MCI, and early Alzheimer's. Again, it's amazing that we actually can't ever see any differences with the treatments that we use to use these measurements to say that treatments actually work or not. So, why, what about digital assessments improves that opportunity, that difficulty? And I think one of the basic principles is that, we know that the conventional approach of testing people intermittently gives you these data points, then we apply some kind of analysis and say, oh, there's a trend, but then we might get some other report that, oh, there's some symptom that has popped up. And then we're challenged to really relate that data to that symptom report. Imagine if you have data that's every day or continuous. What you would realize in this example that there's actually no real mean change. But in fact, there is a change. And the change that might be observed is change in the variability over time. And that's something you can only get with digital assessments and measurements that are occurring frequently. And that change itself may be a marker for early detection, for example. There are many other advantages. I don't have time to go into all these, but I sort of alluded to them in the beginning here. The fact that these measurements can be done inobtrusively, ambiently, that they're real time, they're time stamped, so you can look at data that's holistic, and knowing how one type of assessment relates to another is very important. And the bottom line, they're ecologically valid, and therefore really are looking at, you're able to look at meaningful change. Now there are a blizzard of, northern term, of assessments that are available. We tend to focus on methodology, and I think this is going to be one of the things that I would like to emphasize in my brief time, that it shouldn't be about the technology, it should be about what we're trying to assess, what's the problem we're trying to address. A lot of times it's like I have a technology, how can I use it, as opposed to I have this question, what is the best technology to do the assessment? And so there's tons of wearables, many thousands of apps and devices that connect to those apps, many kinds of passive sensors. And then the mother of all things is really this integration into sort of the Internet of Things approach. So there are many, there's a lot of promise here and many approaches, but there are admittedly still a lot of challenges. We've taken an approach of, trying to be really use case directed and thinking about not the technologies per se as first principle. So what you see around this home are a bunch of technologies or devices or sensors, but really the most important thing are those subcategories of function. So cognition, mobility, sleep, socialization, health behaviors, I'm going to read them all, safety, physiology, environment. These are all major domains of functional assessment, if you will, that are really important. And that's also this platform that we've developed is technology agnostic. It's really about what devices work best in a particular situation. We also incorporate the legacy kinds of data, the EMR data, the research data, the fluid biomarkers, the imaging, whatever, and other external information such as weather data. All of it is developed into databases, and I won't have time to talk about that, except at the very end. I want to emphasize that. The other point I want to emphasize is that technology is changing all the time. In the current cycles of funding, by the time you've written a grant, submitted it, and it's awarded, so now it's maybe 2 years later you get a notice of award, the technology's maybe gone, in fact, and there's something very better. So you have to be available, ready to use something new or something better, because these things change all the time. But you also have to be able to relate to what was being done, what has been done in the past. So I want to focus now on just some context of use, some examples. There are multiple areas of assessment traditionally. I'm going to divide these into prediction, diagnosis, and monitoring, and just assessment methods.
Thank you so much. It's such a pleasure to be here today. So that was the Buck Institute. At the Buck Institute, we're really trying to understand aging. And then not only aging mechanisms, but really how that relates to brain aging, neurological diseases, and other diseases that may result as part of aging, and where aging is a risk factor. So I'm listing a few here: neurodegeneration, osteoarthritis, kidney disease, cancer, radiant aging. All of those are things that elderly people suffer from. Here today we will focus a bit on the neurodegeneration, but also on the link to cellular senescence. We talked about molecular markers earlier, that this is something we can also do. So I wanted to show you the technologies we're using, mass spectrometry. I wanted to show you special days in my life, installation days. So these are our instruments that work with us. These are really very highly sophisticated instruments that can do molecular measurements. We've been really lucky at the Buck Institute to continuously upgrade our instrumentation to stay at the state of the art, to do molecular measurements in difficult systems of biology. So you can see about every year or two we update our instrumentation, and the other ones continue to work, and now more recently we also started doing metabolomics in addition to proteomics. Yeah, those are always exciting days. Now what can we do with all this technology? We can really try to kind of understand what are some key mechanisms of aging. One big focus at the Buck Institute is clearly cellular senescence, the state of cells where they stop dividing, and we do really think that they play a big role in neurodegeneration also. But other hallmarks of aging we're also investigating. Cellular senescence is really something where cells stop dividing and all of a sudden they secrete all kinds of things. This goes into the microenvironment of tissues, but also into circulations. So one idea is really, can we maybe find things that eventually, are a molecular signature, something goes wrong in the tissue or organ that we can find in biofluids such as plasma or cerebrospinal fluid. We can look for soluble proteins, metabolites, or exosomes. Typically, one thing we do is we discover something in the original tissue or organoid or cell type, and then look for that in the circulation, which is much easier and a little bit hypothesis-driven, even though there's an underlying unbiased approach behind this. And so this is really a big power. And then being able to monitor in biofluids, for example, changes between young and old, between kind of healthy and ADRD, and also monitoring treatments and the efficacy of the treatment. We also talked about the value of longitudinal measurements. So this we can do all these things on a molecular level now, based on in difference to the first session where it was more the phenotypic, and then integration of both of these. And so we actually showed that some of these senescence markers we were able to find in human aging studies at the Baltimore longitudinal study, so that was a proof of concept. And our idea is now to do the same and try to find this not only in aging, but also in neurological diseases and maybe finding early markers. One thing we have really focused with Lisa Ellerby and the team, and also Tara Tracy, who's also here, is looking at risk factors, genetic risk factors. We talked about ApoEs, those are apolipoproteins. We know that most people have the ApoE3 allele, but you know, ApoE2 is thought to be somewhat protective or resilient, and then ApoE4 has an increased AD and ADRD risk. And really, these are small molecular changes, but they really very dominantly impact lipid binding, and this has huge effects on many different things that can happen in the brain: synaptic deficits, neuroinflammation, impaired glucose metabolism, or tau-mediated neurodegeneration, integrity of the blood-brain barrier even. So we're trying to learn about this, and then research questions that we're asking first to then develop it into biomarkers are the following. For example, we think that there's evidence of senescent cells in brain that may drive AD. How does all of that also is influenced by ApoE genotypes? Does that influence the detriment of senescent cells? We can measure this in organoids or cell types. And then, you know, what particular brain cell types are more susceptible to senescence and disease? And you know, with Lisa Ellerby and her team, and actually Tyne is here today, Lisa has derived from iPSC cells astrocytes, neurons, microglia, pericytes, and cerebral organoids. And those we then use to develop markers that later we want to look in these patients to stratify patients for early development of disease in the brain. And so, you know, we developed these organoids in Lisa's lab, you know, over weeks and months indeed, for these different genotypes of ApoE2, 3, and 4. And then we're asking how are they, you know, resilient or affected to senescence, you know, implicating early or accelerated aging. And you can see that these are long experiments to be done, often over several months. And then all of this comes to my lab where we're doing these molecular measurements. So it's always really exciting. And so now I hope I can show you a little bit what we do from here. So when you look at, for example, this is like what these organoids actually secrete into the environment. You can see we monitor maybe here 900 proteins, and everything in red goes up and everything in blue goes down when we compare an ApoE2, the senescence versus control. It is interesting that these different genotype organoids all have somewhat different profiles. So they all have different proteins that they secrete. And when I say secrete, what should you think about that? This is from these cell types, these organoids mimicking a human brain, maybe in the dish. It changes the microenvironment and maybe makes it more prone for disease. So understanding that is really relevant for mechanism, but again, as I said, also for biomarker development. What are some of these proteins? Looking here at the ApoE4, which has the biggest risk factor for Alzheimer's and ADRD. So those are proteins here, I'm listing a few, but just pointing out some of these calmodulin proteins have been linked to Alzheimer's, and particularly some of these proteins with the S100 are really relevant also. So kind of looking a little bit, what are the candidates that we could maybe get from our cerebral brain organoids, and then we would actually love to, with Caitlin's many data of homologic proteomics on human large cohorts, I would love to compare, can we actually stratify how we mine large cohort data that already exists here at your center? So I think that would be actually a very, really interesting next step. So for example, looking at which proteins are more prone in the ApoE4 versus the 3 and 2, again, we highlight with the red dots the ones that are significantly regulated in the different genotypes. And again, these S100 proteins show up. They have been described in Alzheimer's disease, and they're really important. They have an effect on tau biology, neurofibrillary tangles. They have an effect on the APP processing, which could result in amyloid beta aggregation, and also in zinc homeostasis, which can actually be protective but also detrimental. So I feel with our unbiased approaches, we can give hypothesis ideas how we can mine all the already existing data that you all have here, but then at the same time also maybe develop specific assays for specific targets that are really fast and easy to do. So I do think this mass spectrometry can be a great hypothesis builder, but also understanding really the mechanisms behind all of that then could lead to drug target development. In fact, in Lisa's lab, they do this often, what drug targets could modify these pathways. So you can use, again, mass spectrometry or other proteomic technologies then to bring this into candidate biomarkers to stratify patients. But I wanted to, it's actually interesting, Jeff, you mentioned faults in patients with cognitive decline, and I mentioned this a few years ago here at a grand rounds with Joel and Bruce Miller. So I want to bring up something kind of maybe you haven't thought about, but maybe you have. Well, we all think about neurons and axons and dendritic structures and networks of neurons. You can see a little part of the brain, the neuron portfolio, and then the extended outreaches that these neurons form. Osteocytes, cells and bones do the same. They have huge lacino-nuclear networks, cells in the bone. The bone is a living tissue. And as the human system ages, there continues to be bone-brain communication through the circulation, through exosomes, through all kinds of factors that maybe we don't understand, particularly not as much from the bone to the brain, maybe more from the brain to the bone. And as there's aging, this becomes more detrimental and can inform each other. In fact, we find, this is my own data in my own lab, that the highest proteins found when we do proteomics on bone, which as you can imagine is difficult, are proteins that we find and think of being in the brain. ApoE, ApoA1, APP, amyloid beta, and so that was interesting. There have been huge epidemiological studies finding that OA patients are more likely to develop cognitive decline, and those are huge studies with big patient populations, and the other way around. So, even controlling for all the things one has to control for, AD patients are more prone for osteoporosis and hip fracture. So, the concept is interesting to think about this protein-protein interaction. So now, how can we assay this with a molecular assay? So the question, for example, is at the back, Lisa Ellerbici and also Tara again, they have these mouse models of neurological diseases. Well, at some point I was at some conference and somebody told me, when we have these Alzheimer's mice, their skull is more brittle. I'm like, whoa, that's interesting. And why is that? So there are actually phenotypes. And so we're asking in these ApoE2, 3, and 4 mice that have humanized ApoE, are there differences in the bone health? Maybe are the bones less healthy in an ApoE4 mouse? Those are kind of interesting questions. But, we go into this and we don't know the answer, right? But that's the science behind it, right? Trying to find that out. And it is amazing, some of the things we did find, how differences are. So for example, this is the ApoE4 allele mouse. compared to an ApoE3 mouse. So again, ApoE4 more risk for AD versus what most people have, the ApoE3 allele. You can see huge changes in this volcano plot. So many proteins upregulated. Out of almost 2,000 proteins we see in the bone, this is the bone, we see about 400 proteins that are differentially regulated in the ApoE4 mouse over E2. So maybe, we think in Alzheimer's and neurological diseases always about the brain, but are other organs systemically affected by these types of diseases? And this definitely suggests this. Pathways that we find have to do with neuroinflammation, mitochondrial dysfunction, and even sirtuin signaling or integrin signaling. But what is interesting, I want to show you in the next slide. When you look at this, this is in female mice in the bone, comparing the different alleles to each other, always comparing to E3, and I want to focus on the middle panel, which is ApoE4 to ApoE3. In the bone, you have huge changes. In the brain of these mice, we have really nicely significant changes, colored in red upregulated and blue downregulated, and that is really relevant and important. But I just want to point out that in the bone, we also see a multitude and an immense amount of remodeling and changes. So my bone collaborator, Tamara Elliston, actually said Alzheimer's is really a bone disease. So, but I just want us to think a little bit outside the box, that there is this organ-organ communication. So I wanted to, with my talk, really open the door of really having molecular measurements in neurological diseases, but also not just measuring and not just looking what is in blood and finding from there, but maybe taking hypotheses, what we can do in the lab to inform what we look in patient cohorts. I would love to integrate with some of the earlier session data that we had. And we know that aging signatures are also still in neurological diseases, so maybe aging signatures could even be early molecular markers, but also we're looking for these disease-specific markers. And I talked about the bone-brain axis, we call it like that. And really, in the end, one of our goals is always to go into biofluids. Thank you so much. This is my lab. I want to point out this is Joanna Bonds here on the next to the fountain, and Charlie Schumann, and this have been big collaborative efforts. I'd love to work more with you on this particular topic. Thank you.
Well, Birgit just teed me up quite nicely. She talked about proteomics, she talked about aging, she talked about biofluids. So I don't have a lot left to say, although I will try my best to answer your call to integrate some of these methods in patient cohorts and patient outcomes. So that's going to be the focus of my presentation today, is using CSF proteomics to identify the biological mediators of the effects of age on memory. So this diagram on the right represents some of the most common post-mortem neuropathological measurements that we measure in our human studies. So when patients go to autopsy, we can measure these different proteinopathies. And these have largely, for good reason, been the focus of research in neurodegenerative disease. These are proteins that we use to define pathologically these diseases. But when we look at the relationship between proteinopathies measured at autopsy and cognitive function during life, we observe that less than half the variance in cognitive function in life is accounted for collectively by postmortem pathology. And so this really stresses the need to understand additional molecular sources of cognitive aging. So one way we can think about answering this question is understanding some of the very clear established factors that relate to cognition, in this case, age and sex. So these are cornerstones of cognition, cornerstones of episodic memory. The data presented here from the Baltimore longitudinal study of aging, these are all clinically normal adults. And what's readily apparent is that with age, you see a consistent decline in memory. And there's also some pretty robust sex differences in episodic memory, a well-established female verbal memory advantage. For my talk today, I'm going to focus just on aging due to time, but all the models and data that you see, we've run in parallel, answering some questions related to sex as well. So the tool that I'm going to talk about today is large-scale CSF proteomics, and this allows us to have a real-time window to the brain. This study had patients with Alzheimer's disease in control, and they did mass spec proteomics in CSF. They also had parallel data in brain, and they were able to detect about 2/3 of the proteins that were in brain were also detected in CSF. More recent studies have been able to actually increase the depth of coverage in the CSF proteome, where I think we're getting even closer. The volcano plot on the right just illustrates that many differences in the CSF of Alzheimer's patients can be detected compared to controls. So, the overarching goals of the project today is to use large-scale proteomics to identify the biological pathways that underlie the established effects of age on memory, and also identify individual targets that can recapitulate the effects of age on memory, such that if you put age into a model, it's no longer significant, and we've actually measured some underlying biology that accounts for the aging phenomena. The data are from our Hilton Brain Aging Center cohort. We had 95 functionally intact, typically aging adults in this study. They did not report any cognitive symptoms, so they were clinically normal, and they all completed a lumbar puncture with CSF collection. And we used an aptamer-based proteomic approach, Somologic, which screens for over 7,000 proteins. And in addition, they complete comprehensive cognitive testing. For our purpose, we focused on a memory composite composed of the individual tests you see here. And they complete brain volumetric MRI. So for this project, we focused on the medial frontal lobe. And just to establish, Caitlin's introductory remarks showed a plot of age and cognition, and here we see a similar pattern. Pretty reliable changes with age. I'm not going to be focusing on the outliers and the juicy heterogeneity that she described. The goal of this project is really to identify what is most strongly representing what we think of the effects of age. So I'll direct you to our analytic workflow. In the upper left corner, what we first did is we looked at what's the relationship between age and the CSF proteome, and what's the relationship between the CSF proteome and memory performance. And from there, we can filter down and identify proteins that are both associated with aging and episodic memory, look at some broader biological pathways that are overrepresented in that shared overlapping list. And then we can move towards a more individual protein approach. Perform targeted mediation models where we identify CSF proteins that actually statistically mediate the effects of age on memory, and then take those biomarker candidates and see if they correlate with an orthogonal measure of brain integrity in medial frontal lobe volume. So the figure on the left is a volcano plot. The x-axis represents the relationship of each individual protein with age. Proteins to the right are increased with age, proteins to the left are decreased with age. And really the take home here is we can detect really robust changes in the CSF proteome with age. This is only a study sample size of 95, and we're able to get over 25% of the proteome significantly linked to age. This slide now represents the overlap between the aging proteome and the memory proteome. So the x-axis stays the same. It's the effect of a protein's relationship with age. The y-axis represents the protein's association with memory. So the upper left quadrant, the individual proteins circled in blue, are proteins that with age they decrease in abundance, and with better memory they increase in abundance. So you could think of these as younger age, better memory proteins. In the bottom right quadrant, we see the inverse. So proteins that increase with age, and as they are increasing, they predict worse memory. So taking those subsets, we can look at pathway enrichment. So for the proteins that increased, that were associated with younger age and better memory, we see a strong enrichment for pathways linked to chromatin dynamics and genomic stability. So we see gene ontology terms related to DNA metabolism, chromatin, and DNA repair and binding. When we look at the proteins associated with older age and worse memory, we see strong enrichment for complement activation, coagulation cascade, proteins involved in innate immune response that are increasing in the CSF with age and worse memory. So now towards our targeted approach, we ran mediation models on those subset lists, and here we identified 7 proteins that significantly mediated the effects of age on episodic memory. The x-axis represents the percent of the direct effect of age that's mediated by each individual protein. Some individual hits that I want to highlight here: complement factor D, which is a rate-limiting enzyme in the alternative complement pathway, was the individual protein by itself mediated over 50% of the effect of age on memory. And then also lipocalin-2, which I saw listed in Birgit's slides as an osteokine. Here measured in CSF, we were able to identify lipocalin-2 and LIL-RA5 as important innate immune proteins that mediated the effects of age on memory. So focusing on lipocalin-2 and LILRA5, we see that controlling for age, sex, intracranial volume, they higher levels predicted lower volumes of medial temporal lobe. Again, supporting the relevance to neural systems that support episodic memory with aging. So with that, a few take-homes. So large-scale proteomics in CSF is a powerful tool that we can use to identify pathways and individual targets that link age, and I didn't have a chance to show today, but sex as well to episodic memory. One particular pathway that emerged as being important for the effects of aging on memory was innate immunity. Like complement factor D, as I mentioned, it's a rate-limiting enzyme in the complement pathway. We know that the complement pathway is very important for tagging synapses for elimination and just restructuring synaptic structure. Lipocalin-2 is secreted by reactive astrocytes, and it's a pro-inflammatory marker with known neurotoxic effects. It's been studied in other conditions like HIV and traumatic brain injury. And LILRA5 is a macrophage marker that's enriched in the correlated plexus stroma, so very close to the CSF system, may be very easy to detect in CSF, and has also been linked as a genetic risk factor for late onset AD. We also observed a few other pathways, including those linked to genomic stability. And with this discovery-based work, I agree that it's so important for us to engage with our preclinical folks who understand these pathways deeply and can rigorously test these different targets in their experimental models. So that's one future direction that we'd like to take. Again, from our approach, there's no need to imply causation here. These data are cross-sectional, they're observational, but important to just take into account longitudinal data really critical. We want to look at other outcomes beyond memory, but we use that as an anchor point for a cognitive ability that is declines with aging and the importance and relevance to neurodegenerative disease. And so we'd like to apply these age proteomic signatures to disease cohorts, validate on other platforms like mass spec, and ultimately, for clinical scalability, understand what measures in the periphery can be faithful indicators of what's going on in the brain. So with that, I'd like to thank the Hillbloom Foundation. My first job in clinical research was coordinating this Hillbloom study, and so it's been a really rewarding to come back and work on some of the data that we collected even 10 years ago. Big shout out to Joel, Caitlin, and the rest of the aging team and other sources of funding as well. Thank you.
Hi everybody. I'm going to talk to you about PET today, and I'm titled my talk Scaling the Gold Standard. And I'm going to talk about how we use these biomarkers in heterogeneous multicohort samples in our lab. So it is, I think, widely accepted that PET is the gold standard to measure amyloid tau in the brain. This image shows representative amyloid PET images on the top row and tau PET images on the second row, with increasing abnormality from left to right, from normal to what you might expect to see in someone who's dementia on the far right. And rather than spend my 10 minutes talking to you about why we think amyloid PET can accurately measure amyloid in an individual, I'm going to talk a little bit more about some of the limitations of PET imaging and how we in the lab are addressing some of these limitations. So compared to fluid biomarkers, tau PET gets you spatial photography, spatial information of insoluble, accumulated pathology. It's better for staging and in comparison to neuropath, and it's further along in standardization in terms of cross-lab, cross-scanner standardization work, but it's often called out for being less scalable because it's costly, it's invasive, it takes a long time to collect a PET scan. And another biomarkers is the question of generalizability and reproducibility. So, are our thresholds and findings actually generalizable and reproducible to other populations that we know are not generally engaged in academic research? So, this issue of generalizability is really a big area of focus in biomarker research right now, because we understand that every particular study's cohort is going to be the direct result of that study's approach to recruitment, the sites where they recruit the people, the participants, and observational studies, clinical trials will reach different groups of participants. And we also know that there are some groups of participants that are not being reached at all by any of these different studies, by and large. But for the purposes of my talk today, I'll just highlight some of these differences across some of the cohorts that we work with. So first, the Berkeley Aging Cohort Study. This is a longitudinal observational study of a convenient sample of older adults that are mostly white and highly educated and live around Berkeley. The ADNI sample was designed to be a clinical trial-esque sample of older adults and impaired individuals who have geographic diversity, so they're recruited all across the country, but very low CVD because of the exclusion criteria. The Pointer Imaging Study is a subset of individuals enrolled into a multi-domain lifestyle intervention. They tried really hard to do community recruitment, and so there's higher ethno-racial diversity, higher CVD, and poor lifestyle factors. The SCAN study is an Alzheimer's disease research center-based cohort. So again, a lot of geographic diversity, some ethno-racial diversity, and unique clinical diversity because a lot of these places are tertiary neurology centers. And then they have the HD study. This is a longitudinal observational study, but has a unique design where they've recruited equal-sized ethno-racial groups across 3 different groups. So all of these studies collect amyloid and tau PET, and the raw images have been processed by our group at Berkeley using our harmonized pipeline. And the idea is that if we combine all these data together, which we can do if we analyze raw data in this way, then we have a sample that is more generalizable because they suffer a little bit less from the unique characteristics of each study. And so today I'm going to focus on this critical step in AD pathogenesis, which is the relationship between amyloid and tau pathology. This is the plot of 2,500 cognitively normal older adults, and on the x-axis we have global amyloid measured in centiloids. We use tracer-specific conversion equations for 4 different amyloid tracers to put them onto this common scale. And for tau, we're using a z-score approach where we use reference samples again for each tracer, 3 different tracers, using amyloid negative individuals age 60 to 70 as our reference sample. And you can see there's a lot of variability in this association, and we're really interested in what is driving that variability. So we're going to look at some potential modifiers of this relationship and what might be driving that variability, including sex, which we know is related to tau and tau PET off-target signal, importantly, actually. It modifies amyloid tau associations in the CSF, and women are, of course, twice as likely to be diagnosed with AD compared to men. We'll also look at race and ethnicity. It's been shown that there are lower amyloid positivity rates in minoritized groups, and that CSF tau varies by race. And we'll also look at ApoE, where we've already heard a little bit about E4 risk allele versus E2 protective allele, and there's a relationship here also between race and ethnicity and ApoE genotype. And finally, education, which is essentially a social determinant of health and an early proxy for cognitive reserve and life advantages. And we predict that sex and ApoE will modify this amyloid and tau relationship, and that we'll see larger effects with amyloid positive patients, and we were not sure what we would see with the other 2 predictors or modifiers. So we'll focus first on unimpaired individuals to look at really the earliest stages of AD. our pooled sample has a mean age of just under 70, 66% white, which is better than many cohorts out there. 28% are APOE4 carriers, and our education is less, mean education is less than 16 years. I'll walk through this plot a little bit to show a bunch of them. This is entorhinal tau, or medial temporal tau, plotted by sex and amyloid status, and the shape of the points shows which study this data came from. And we take this raw data and we use a linear mixed effects model where we have a random effective study, a cohort study, and we adjust for age, and we are really interested in this amyloid-sex interaction effect on predicting tau. And we find that women generally have higher levels of medial temporal tau for a given level of amyloid compared to men. We're going to do the same thing now with the other moderators I mentioned. So for race and ethnicity, this is again the raw data plotted by amyloid status and ethno-racial group. And we'll really focus on just the non-Hispanic, white, black, and white Hispanic groups where we have enough data to model them. And we see that there is this effect such that black individuals have lower tau than expected compared to the other racial groups for a given amyloid burden. For ApoE, we see a strong effect of ApoE4 carriage, such that they have higher rates of tau for given amyloid burden. And this effect remains significant even after we adjust for race and ethnicity. Finally, we looked at education, and while there does not appear to be any relationship between education and environmental tau in the raw data, there is a very subtle, small effect size effects such that people with higher education have slightly lower tau for a given level of amyloid burden compared to their peers with lower education. So now we're going to conduct the same analyses, this time with the addition of about 600 amyloid-positive impaired individuals. So if you look at the top row here in red, you'll see the percentage of the total cohort now that for our impaired individuals. And we want to see whether or not our findings remain consistent, and are they really sort of representing early Alzheimer's related changes or relationships in the brain. And you'll note that the E4 carrier rate went up significantly because, again, we're requiring that all the patients are amyloid positive. So this is the same effect we saw before, and you can see it remains that women, there's this sex effect in the relationship between amyloid and tau. And now we're also going to look, because we have more variability in our tau measures, in a slightly later ROI, a temporal meta-ROI, that captures better the whole, from normals to impaired individuals, the whole spectrum of tau burden. And you can see that this effect is attenuated a bit. It's significant, but it's a smaller effect. With race and ethnicity, we did not see any consistent findings. It didn't. The effect we saw in the unimpaired only didn't replicate here, and we're not very confident in anything that we're seeing. For ApoE, we see a very consistent, strong effect of ApoE4 carriage, driving greater tau, given your amyloid burden. And then for education, we also see a very consistent but very subtle effect, higher education being cross-protective in amyloid driving tau. So to wrap up, PET biomarkers are the gold standard. They're used clinically. They are sort of precision biomarkers, I would argue. But they are, and they're also further along in terms of cross-lab and cross-tracer standardization. And we have, because they're more widely available, we do have an opportunity to do large studies like this. We hope to, this is just our first pass of this. We hope to have many more thousands of participants in these kinds of studies in our lab as PET imaging is more widely available and more likely used. We saw that ApoE4 carriage was really consistently interacting with amyloid to drive higher tau burden in all of our models. We found that sex does modify the effect of amyloid in medial temporal lobe, in unimpaired especially, but it's more subtle in the meta-ROI and in impaired individuals. And education was a really consistent modifier of this relationship, but with a very small effect size. Finally, in a model where we can include all four of these modifiers in a single model, they all still have significant effects, interaction with amyloid on tau burden. And if you look at the effects that I showed here in the pooled sample in individual cohorts, you'll find that they are not replicated across each individual cohort on their own, which either be a statistical power issue or an issue with the cohort characteristics themselves. finally, we aim to use some epidemiological methods, inverse odds weighting, to really actually quantify the generalizability of these different cohorts in our pooled sample, using the health and retirement study as our reference data set. And we want to work on better harmonized cognitive data across these scores, so we can also examine modifiers of tau driving cognition. And then finally, many of these studies are actively collecting longitudinal data, and in a year or two, we will have enough longitudinal data to really look at that on a large scale as well. So with that, I'll end and thank the Vegas Lab at UC Berkeley, all of the collaborators involved in running these large studies, and you all for your attention.
I'll speak loudly. Great talks. Specific questions for Birgit and Rowan. So thank you for reminding all of us that we're overly focused on pathology of misfolded proteins and cerebrovascular disease, that there's so much more out there. And you introduced us to a lot of different proteins and molecules. Can you help me out a little bit, and if you can, boil it down to what you think might be the 2 or 3 most important biological pathways that are impacting brain aging, and why are they influencing brain aging?
I'm not going to do that because I think it's more than 3. But to name a few, I think we've heard a lot about immune response, and that can manifest in different tissues in different ways. So that's a very broad umbrella category, and I'm very eager to hear Birgit's thoughts. I think I'll just say when we think about brain inflammation, the complement pathway that I highlighted, I think is very important because of its links to synaptic health. Thinking of synapses, the crux of cognition, and these complement proteins going in and actually affecting the health of synapses in the context of an immune response, I would say is a very important pathway of aging.
Thank you, and good answer and great question. Yeah, so I think I really also am a big proponent of immune response, but I still really like to highlight the cellular senescence. For example, when I looked at one of your graphics with the CSF, one of your way up biggest thing was GDF15, which is a super strong senescence marker. And I saw in your data set several of those, and we see them continuously. I do think that's an interesting mechanism, and that could potentially have some therapeutic intervention possibilities. Even though it's hard to give a senolytic systemically, we should not do that, but one could think about ways. Yeah, I think keeping an open mind to, I think there will never be just one pathway, but yeah, well, if one picks some that one can target. And for biomarkers, I think it's strong to have panels of biomarkers, and you were very close in that. And then I love the idea of connecting it with other measurements, right, like physiological measurements. And so I would be really interested, for example, in if you had blood samples from some of your patients, right, what the signatures of those might look like.
Thank you. To follow up, the theme here, so the Hillblom Brain Aging Center is going to now be renamed to the Systemic Aging Center, because it's clearly a systemic problem. Tessa, I'm curious in terms of maybe looking at the health of the individual. In other words, and you're stuck with what data's been collected and you can harmonize across all these different studies. And I realize, so for example, education probably isn't the best measure of, but it's what you had to look at. But in terms of looking at other medical conditions, there may be some opportunities to generate a frailty index, which, like Rockwood is, just adding up the conditions, for example, that I think would be very interesting, perhaps. And you've probably already done it, but I just think that that's a great opportunity, perhaps.
Yeah, you're completely right that we're a little hamstrung by what we can do when we're trying to, think in the work I described today, our focus is really on generalizability and getting the largest sample we can get, which limits the variables that we can look at. But in that vein, where we've gone first is trying to really harmonize data for cardiovascular risk and health as much as we can, and we have actually across all 5 of these samples created what we are calling like a modified Framingham risk score. You know, we have to but we're doing it the same way across the 5 cohorts, right? So we make it so that we can create a score that is very close to the original Framingham and the same, so we don't have biases from centrally calculated FRSs in different groups. And along with that, a harmonized way of calculating white matter hyperintensities. And then within the Framingham risk score, we have, blood pressure, either BMI or a cholesterol measure. And so once we see anything with FRS, we then break it down and we want to know what's actually driving that association. And so we're moving that direction there. In terms of like lifestyle, it's even harder, right, to get consistent data across these different studies. But when we can do it, the lifestyle and also like early exposure, things like this. When we can do it, we will do sub-analyses. So we do have ADI, like area deprivation index, calculated on several of these large studies. So we'll do like sub-analysis with that as we move towards answering those questions. I agree. Even years of education, though, were not collected identically in all these studies, right? Several of them collected, age, education categories that we had to then, create. So it is the harmonization step is onerous.
Right, and one last very short question.
Yes, I promise.
You're the boss.
That was a wonderful overview of the landscape of biomarkers, from imaging all the way down to molecule. I guess a question for the full panel, thinking about the canonical gold standard pathology biomarkers, we have them in clinic. How do we think about starting to integrate this explosion of omics that might provide other insights into how do we prognosticate disease? How do we integrate these new biomarkers with the canonical biomarkers and start to wrap our heads around that in terms of moving into clinic and precision health?
Thank you. Yeah, I think biomarkers can be so many things, right? They can be early detection, diagnosis, and that may be very difficult to go into a clinic where they have, for decades certain diagnostic tests that they've been using. But one can really also use biomarkers maybe to monitor interventions. And I really see a huge potential in that, because that has been such a difficult thing, right? How do you know that your intervention works? How do you, for example, know that maybe exercise improves cognitive behavior, right? Like, so having molecular markers for some of those things, and then starting implementing markers like that and bringing value to those, and then reaching into the other areas of how a biomarker can be too. So I kind of see a huge value in that. And then still, the biomarkers are also molecular understanding of pathways, right? And really informing best treatments. So, yeah.
Yeah, I think it's a great point, and I think about what Jeff said this morning about context of use. I think that's critical in the context of clinical use. So, like you alluded to, is this a biomarker for measuring a therapeutic response? Is this a biomarker for risk screening? Is this a biomarker for diagnosis? And all different contexts are going to require different types of evidence to actually support their implementation in a clinical setting. For our observational data that we have in our aging cohort, I think a lot about risk screening, right? These are folks that were longitudinally followed. They entered the study without any reported cognitive difficulties, and we want to know what is signaling the earliest signs of cognitive demise. And I don't think an individual molecule is going to tell us everything we need to know. I think we've learned that a lot through the proteinopathy space. But ultimately, can we get a panel of markers that consistently and reliably predicts with a certain level of precision? And that may not be the same in each individual, and that's sort of a lot of the data challenges and the conceptual challenges come, but we need larger cohorts, deeper methods, and consistent methods to be able to better tackle that.
I'll say one thing quickly, which is that I think, Lena mentioned like validation of new markers with old or new tools with old tools. And I think we need to make sure to pay attention to whether or not new biomarkers are explaining new or unique variants in what we're trying to predict, or they're also predicting it in addition to these biomarkers. So I think more head-to-head understanding, yeah, we can have a panel of all these things, but is it redundant? Are we actually basically buying ourselves the same explanatory power with multiple measures? We don't need to do it. So.
Oh, I wanted to bring up one cancer example, right? Where in cancer, it's not only about detecting somebody has cancer, but it's really stratifying what treatment should these people get. Which the best example I feel is breast cancer, right? Where you really know this person would not respond to that treatment, and then you use the other treatment. And I mean, because neurological diseases, ADRDs, are so manifold, I don't think it's just one disease, right? And you all know much better than I. So really using it also, not just diagnosis, but to stratify treatments. And that's where I see it not being redundant, hopefully, and having real value for the patient, right?
Thanks for having me. It's a pleasure to be here. Sorry that it's not Judy. I was actually really disappointed not to be able to hang out with her today. But nonetheless, hopefully this will be entertaining, hopefully as entertaining, because I know she's really fun to listen to and speak with. So anyway, it's my pleasure to talk to you a little bit about how non-invasive electrical stimulation can be used to facilitate cognitive function in older adults, and how we might be able to optimize or tailor that stimulation for optimal benefits. Now, the utility of electrical stimulation for health purposes is not new. It's actually been around since the Romans. They used harness the power of the electrical fish to treat headaches and gout. Now today, we do not have fish in our lab. We use batteries, and it powers devices such as these pictured here, known as transcranial electrical stimulation. Generally, you're passing an electrical current through an anode, into the anode, out a cathode, and hopefully some of that current goes into the brain. Now there's generally three generic forms of TES. One is known as direct current stimulation, where you're passing the current straight from anode to cathode, right through the brain. There's alternating current stimulation, which I'll be focusing on today, where you choose a frequency, hopefully associated with some sort of cognitive function that you're targeting, and that just oscillates between these electrodes, 2 or more electrodes. And then there's random noise stimulation, where you basically blast a whole bunch of different frequencies and hopefully something modulates the system in some meaningful way. So as I said, we're going to focus on TACS. So I'm going to talk to you about 2 different frequencies that could be used to target different cognitive functions. I'm going to start with theta band, or 6 Hz TACS stimulation, and how it could be used to modulate cognitive control in older adults. When I'm talking about cognitive control, I'm going to specifically talk about multitasking. It is our ability, or lack thereof, to do 2 or more things at once. It's been estimated that upwards of 97% of the population try to, but cannot, multitask, and that results in upwards of a 40% reduction in performance. So the take-home message here is it's better to do one thing at a time. Now, there is a real-world impact for multitasking, and that is why it's illegal to text and drive in 49 of the 50 states. If you're wondering where that last state is, it's Montana, so have fun. Enjoy it while you can. It's illegal in some counties and some municipalities, so don't go too rogue if you visit. That being said, some colleagues of mine were interested in the impact of aging on multitasking performance, particularly in this paradigm here that we lovingly refer to as neuroracer. In one condition on the left, you see a sign, a green circle, that is your target. Ignore all other shapes or colors. In the second condition, where it's another single task paradigm, where you just drive a car using a controller, just try to keep it on the road, ideally. And then, of course, there's the multitasking condition, where you're driving the car and responding to these street signs simultaneously. As you probably would have guessed, multitasking performance declines as you get older. There's a greater cost associated with it. Fortunately, the good news is, with training, with practice, you can improve that ability to multitask, and those improvements sustain for, at least in this paper, is sustained for upwards of 6 months, although they recently published a paper showing that those effects sustain for 6 years. Now, at baseline, the main neural differences that they saw And this is EEG data. So if you're not familiar with it, it's topography, sort of like looking down on top of the head from a top-down point of view. The top, that little carrot, is actually supposed to be a nose. So anyway, the difference in these aging groups we've noticed was younger adults showed more midline frontal theta activity. It's largely around 6 Hz oscillatory activity during this task. Fortunately, after training, we saw a large increase in this frontal theta activity in older adults. And we localized that to the medial prefrontal cortex. So then the question is, can we sort of shortcut that? Can we actually facilitate multitasking abilities by applying a current at that frequency? So we did this experiment, a series of experiments really, I'm just going to sort of briefly touch on, where we did the same paradigm, this multitasking paradigm, and we did this, our first 2 experiments were in healthy young adults, just as proof of concept, and then we moved over to healthy older adults. And one group received this theta stimulation, full dose, which was about 27 minutes of stimulation over the course of 1 session. And our control group received a much smaller dose, a 5-minute dose of the same frequency, same intensity. And then we just look at multitask performance, specifically focusing on D prime, a signal metric of discriminability to these targets. And in the bottom right corner is our electric field model of how much current we believe got into the brain. And if you're curious, we put electrodes right here at F3, F4, if you're familiar with the 1020 EEG electrode coordinate system. And we just pass the current straight through the prefrontal cortex. Now, as you can see here, these black dots represent our group that received the full dose of theta stimulation, whereas those white dots represent our control groups. And in both cases, in both different healthy young adult groups, we were able to replicate this effect that, yes, frontal theta stimulation is able to facilitate multitasking ability within a day. And then we replicated this again in healthy older adults, age 60 to 80. Now, we weren't just interested in seeing if these results extended to older adults, but we were also interested in whether there might be cumulative benefits, additional gains that may be had by additional days of stimulation. So we applied stimulation 3 days in a row to see if we could get greater gains. And as it turns out, no, we cannot. Those are largely parallel lines. There was a group effect here, but it just did not widen that difference. So it really seems to be operating sort of on a learning curve, at least right here in this example. And it wasn't just specific to D prime, we also showed significant improvements in response time, and those effects sustained for upwards of a month. We didn't test any longer after that. Now, what's interesting here is the individual variability. Not everyone responded to this stimulation, as is much research. Not everyone shows the effect you're looking for. In the bottom right corner, you can see these are essentially electric field models of how much current got into the brain and how that EF correlated with changes in performance. And as it turns out, if you take a frontal lobe mass, mind you, this is electrical currents going through the brain, so it's kind of going everywhere. You saw in the earlier model, it's not very precise, which is a blessing and a curse. We can talk about that later. But nonetheless, the amount of current that got into the prefrontal cortex correlated with the improvements in performance, such that those people who got the most amount of stimulation in the brain were the ones that benefited most from it. So I think that makes a lot of sense. And that's not our only source of variability. Now that variability comes from their neuroanatomy, right? The amount of current that's getting into the brain is contingent on how thick your scalp is, how thick your skull is, how much CSF you have between the dura and the cortex. In addition to that variability, there's variability in neurophysiology. So at baseline, on average, people have these frontal theta activity. On average, it's around 6 Hz. So we applied stimulation at 6 Hz. But not everyone has a peak at 6 Hz. Some people are peaking at 5, some are at 7, some are at 4, some are at 8. And as it turns out, those people who exhibited baseline neural activity before stimulation was applied, those people who exhibited neural activity right around that 6 Hz stimulation were the ones who benefited from it. If you had a peak around 5 or 7, then you benefited less. And those that were peaking, around 4 Hz, 8 Hz, they benefited very little, if any, at all. And if you take that neuroanatomy and neurophysiology, if you take that variability together, it can account for upwards of 65% of the response variability to the stimulation. Well, that's all good and fun, but it doesn't have any sort of clinical efficacy. Can we actually move this into a population that may have more need? So in our next study, we looked at older adults with multi-domain amnestic mild cognitive impairment. And we had a few other questions that we were interested in. One, could they do it to themselves at home? So we gave them a device that operated very much like a Band-Aid. You stuck it right to your head, and it's already pre-dosed in terms of intensity, duration, and frequency. And they just had to press a button, and it applied the stimulation. We gave them a series of these patches, so they were able to apply stimulation to themselves on multiple days. 5 days in the first week, and then a weekly booster session over the course of the next month. And then we assessed not only their multitasking abilities, pre and post after a week, after a month, but we were interested in a potential transfer of benefit. One thing I didn't have time to really talk about is that our original study, where the older adults were just doing cognitive training, they also saw a benefit of not just multitasking performance, but that was associated with increased sustained attention. Because in order to multitask, you really do need to focus your attention for long durations of time. So, as it turns out, multitask performance improved in both groups. We didn't actually see an interesting interaction effect. And that's likely because of practice. We saw before multitasking performance improved due to the stimulation after the first day, but they didn't get greater benefits after 3 days of stimulation. Here we're giving them 8 days of stimulation. And we believe what happened is that the control group caught up, they had plenty of practice. But what's interesting is that sustained attention, the transfer of benefit, was only observed in our group that received TACS. So yes, we believe this theta stimulation can be used to facilitate cognitive control, and I am going to move on to another form of stimulation. It's the same device, just a different frequency, different electrode positions, and it can have various variable different effects. So I'm going to try to go through this relatively quickly, but as you're well aware of, some of the most common hallmarks of Alzheimer's disease is decrease in episodic memory and increase in amyloid plaques. And also, you may not know, you probably do if you do a lot of EEG, is that in aging, particularly in Alzheimer's disease, you see a decline in gamma activity, gamma like around 40 hertz neurostimulatory activity. And so we want to know whether this gamma stimulation could actually have some sort of benefit and potentially even alter AD pathology. So first we did this in a mouse model of Alzheimer's disease, a 5x mouse model. We had 3 different groups, a control group, we had a low dose and a high dose, low intensity, high intensity of stimulation. We dosed them 4 times over the course of a week, and then weekly booster sessions over the course of the next month. We assessed episodic memory, as well as markers of amyloid and microglia activation. The green color indicates staining for microglia, and as you may guess, the control group is on the left, the high intensity is on the right, and we saw essentially this sort of dose-dependent effect in activation of microglia in terms of cell count, body size, and process length. And as you probably know, microglia help clear amyloid, and so we saw a dose-dependent effect in lowering amyloid plaques, such that higher doses result in less amyloid accumulation. And then we assessed episodic memory through the Morris-Water maze, and again, the high-dose group showed the greatest improvements in their ability to conduct the Morris-Water maze, which is our index of episodic memory, and that was also not just their testing sessions were improved, but also their learning sessions were improved. They actually showed a steeper learning curve, so we improved their ability to learn the task and to perform the task. Now, the last thing I just want to mention is a follow-up to this. In humans, this is a single-arm study, small n, so everything I'm about to say, take with a grain of salt. But nonetheless, I think there's some really interesting results here, and I think reason to keep pursuing this line of research. So we basically modeled emulated the mouse study. We provided stimulation 5 days in the first week and then weekly booster sessions for the following month, and we assessed their episodic memory as well as resting state MRI, pre-post stimulation, and we got some blood plasma measures. What's interesting here is that we did see an improvement in episodic memory in these older adults with AMCI. This was indexed by improvements in the short delay cued recall of the, short delay free recall of the CBLT, as well as a verbal improvement in verbal fluency. And then in terms of the resting state data, we saw an increase in functional connectivity within our network, our a priori network of episodic memory, which include the IPL, hippocampus, and Transcranial. And we only saw a change, though, in the IPL to hippocampus. Well, what's cool, I think, is that that change in functional functional connectivity was correlated with change in episodic memory, such that those who showed the greatest increases in functional connectivity were the ones that showed the greatest improvements in episodic memory. Also, those with the greatest increases in functional connectivity showed the greatest increase in hippocampal excitability. And we measured excitability in the hippocampus using magnetic resonance spectroscopy as the ratio of glutamine to GABA, so your excitatory to inhibitory neurotransmitter. And we also saw that that change in functional connectivity was also related to baseline fractional anisotropy in multiple white matter tracts, such as the arcuate-fasciculus, the fornix, the uncinate, and the superior longitudinal fasciculus, indicating that perhaps better white matter integrity, it helps you benefit from this stimulation. Finally, I'm just going to note that also the amount of current that got into the brain, particularly in our regions of interest, in our episodic memory network, the amount of current that got into those regions was related to those performance benefits. So those who got the most stimulation into the brain, again, were the ones that benefited most from it. So yeah, we believe that this gamma TACS can also facilitate episodic memory. So what I've tried to show you really quickly is that TACS can be used in a frequency-specific manner to target multiple age-related cognitive deficits. These effects are variable from person to person, but they do seem to be contingent on your neuroanatomy and your neurophysiology. And fortunately, what I like best about this technique is that it has high tolerability and potential convenience of in-home use, which I think makes it a potential powerful therapeutic to combat cognitive decline and hopefully dementia. So I just want to thank all the people that made this happen, and of course the funding agency. Thank you.
I did my PhD here at UCSF, so this is like coming home for me, a very long time ago, across the quad when there was only one building on this entire campus, to give you a sense for what that was. Birgit very nicely introduced the Buck Institute. This is the Buck. If you've never been there, it's about 20 miles north of Golden Gate. Come visit us. My lab is there. We are really all focused around trying to understand basic mechanisms of aging. And the goal there is truly because most of the diseases of the modern world are essentially your number one risk factor is your age. So if we can target the underlying mechanisms of aging, we can target all these age-related diseases together. And as you can tell, I've really drunk the Kool-Aid. I truly believe that this is an important and really the best way to think about healthy aging. So my lab is really interested in studying how changes in homeostatic circuits with age can drive different phenotypes and systemic aging. So I'm going to attempt something very, very ambitious in 10 minutes. So stick with me. Normally I would give one talk or the other. My science talk, the stuff my lab works on, or the stuff that we work on in the Center for Healthy Aging Learning. In the spirit of collaboration, I want to tell you about my lab, but I also want you all to know about the center because there are resources there and potential collaboration points with other people at the book. So, it's not multitasking, it is in tandem, okay? But there will be a quiz. So I just wanted to point out that our goal is not to extend lifespan, our goal is to extend the number of years that someone's healthy or health span. And I'll come back to why this is important. The part of the brain that I focus on is the hypothalamus. So this is a small structure at the base of the brain. It is really where all of the circuits that control homeostatic systems listed here are located. And I think about it as the master regulator for systemic aging. And that is because with age, there is an increase in inflammation, specifically in the brain, but it doesn't happen everywhere uniformly. It happens in hot spots, and hypothalamus is one of them. And so the kind of things that we see, the phenotypes that we see with normal aging are things that are listed here, and these are all disruptions of homeostatic circuits. Now this is also a really cool part of the brain because these neurons make and use tons, dozens of neuroactive peptides, which I'll talk about in a second. And these are, I think, the kinds of things that mediate intra-tissue communications, coming back to this idea of systemic aging. And I won't go through all the details, but suffice it to say, neuropeptides control not just a whole host of really important physiology, but also the associated behaviors. And so when you're talking about circadian rhythms and circadian circuits, they also control sleep. And when they get dysregulated, it can lead to a lot of different diseases and disorders. And there are literally hundreds of bioactive peptides. And they are the most common signaling molecule in the CNS. I consider them the dark matter of the nervous system because we really haven't been able to study them. So if you don't remember anything else about what I work on, these are like the brain's Wi-Fi. So they mediate communication between cells and tissues that are not physically connected. And they essentially give your brain the ability to direct what's happening in the rest of the body. And this kind of communication is two ways. The brain is something like dictator. It's constantly listening to and integrating feedback from different systems in your body. And neuropeptide is really a misnomer because they can be made and sensed by non-neuronal tissue. So this is, the basis for my entire research lab, which is that disruptions with age and homeostatic signaling from the hypothalamus really leads to systemic aging. If we could understand which peptides are changing in which directions, there's a lot of different potential intervention points. So my lab spends a lot of time building tools to study neuropeptides, and for the sake of moving on to the Center for Health Aging in Women, I would just say that Birgit introduced mass spectrometry really nicely, and what they focus on in Birgit's lab is primarily proteomics, so measuring proteins by mass spec. We do a huge amount of peptidomics, so measuring those bioactive peptides in the cells and the tissues that they come from, because neuropeptides are processed out of larger precursors when they're made. And what that means is that you can't tag them. And so an mRNA doesn't really relate to the levels of active peptides. You really need to measure them, and mass spec is the only way you can do that. So peptidomics is as different from proteomics as lipidomics is. And so this is something where I would love to collaborate with people who have biofluids, especially in humans. We do also build a lot of tools to study peptidergic signaling because they're long-range signals. It's really difficult, actually, to measure them. And I will stop here and tell you about one study that you can use as a proxy to understand some of the stuff that we do in my lab. And this is to ask whether, at the level of neural circuits, can we figure out, if we know what the circuit looks like in a young person or a young animal, can we measure how circuit activity changes with age, and then target the nodes that are changed with things like optogenetics to restore circuit function and thereby restore the physiology. And we use a lot of different systems neuroscience tools to do this. And so Hyun Jang, who's a postdoc in the lab, applied this idea to the fluid homeostasis circuit, which is really important. So as older people are chronically dehydrated, the fluid homeostasis circuit is well described in mice by dozens of labs and also by our collaborator in my lab here at UCSF. And so the question was, could she figure out how the circuit was changing with age and then restore the circuit function and thereby change the behavior. Now, there's going to be a quiz, so I hope you're really paying attention here. But the answer is yes, and I don't expect you to take in any of that. This is available as a preprint, and so you can read about it. But a big takeaway is that, and a broader implication from the study is that during aging, not everything goes wrong in the same way. There's a lot of sex dimorphism in how circuits change, and we can actually target those changes in circuit activity to restore things like behavior, in this case, drinking behavior. Okay. Coming back to this figure here, which really focuses on what we do at the Buck Institute, there is a system that falls off this curve pretty dramatically. This is organ function over time. And you can see it's not totally coordinated, right? Your organs are declining in function over time. It's not exactly the same rate, but what we can say is if you're lucky enough to live to old age, most of the decline will happen in the last few decades of life. And so actually 5-6 years ago now, we were studying a peptidergic system that changed with age and trying to figure out where it was acting, what the mechanism was. And it took us to the reproductive system. And this brought up something that I had never thought about before, but that basically the female reproductive system is aging prematurely. It ages faster than the rest of the tissues in one's body. And while women do live longer than men on average, they spend a significantly longer period of their lives in poor health. And this is directly because of aging in ovaries. So ovaries are aging at about 2.5 times the rate of the rest of the tissues in the female body. And this has dramatic implications for overall health. And I want to say, ovaries are for more than baby making. So they're endocrine organs. They are sitting at the center of a signaling network, much like the brain, where they're talking to almost every tissue in a woman's body. And what that means is that when ovaries aren't working properly at any age, in a 10-year-old female or in a 90-year-old female, it uncovers different health risks. And the health risks that it uncovers depends on the age and genetic background of the person that's impacted. And so while menopause and fertility issues are downstream consequences of ovarian aging, there's lots of other things that are important here. And so I call them the architects of health, and they also set the rate of aging in female bodies. And so everything that we're doing in the Center for Healthy Aging in Women is to reframe the narrative around women's health, to think about things through the lens of ovarian function. And unfortunately, we don't know much about how ovaries work, and so we're coming from a place pretty far behind. This is our center. I won't go through all the people here. I'll just flash up the URL at the end, but it's a mix of internal investigators and external. And our goal truly is to map out ovarian function. You don't need to read all of this, but I wanted to put it out there and say that we are really trying to think about ovaries as the master regulators of aging in female bodies, to understand how they work, and then to use that as a way to extend female health span. We have an external global consortium arm that we call productivehealth.org, and it funds grants to scientists all over the world. We've built resources like an ovarian biology core facility at the Buck Institute to lower the barrier to entry for people who are coming from outside this field. So if you are interested in working on ovarian aging, because it's obviously the most important problem we could tackle in aging research, we've got a core facility that can work with you to lower the barrier to entry for you to start doing experiments in this space. We also have an international conference on reproductive aging, and we spend a lot of time building resources for the community. So if you're interested, you can go to our website. Last but not least, coming back to my lab, we spent a lot of time mapping out the ovaries across time and space, both through an estrous cycle in mice and across aging, using single-cell spatial transcriptomics. And I won't go through all this data, but just want to show you this beautiful, it looks like art, but these are all data from 300 oocytes, 653 follicles, 234 corporal lutea. We learned a lot from this data, and you can read about this in this on biomarkers. Finally, I know that was a lot. We need ambassadors. We need people to help us spread the word about what we're building, the center and the global consortium. And we're also looking for donors right now to help us build. It's really not about ending menopause or changing fertility. It's about really understanding how female bodies work so that we can sync up aging in ovaries with aging in the rest of a woman's body, the same way that reproductive organs are synced up with aging in the male body. So it's really about changing that curve and thinking about how to extend healthy aging in women. So these are the funding sources and also the people in my lab whose work I briefly alluded to, and my collaborators. That's it.
We're going to talk about how aging activates the silent X chromosome in the hippocampus. So, as you may have heard, women live longer than men, and this pattern is really remarkable across the world in literally every country that has reported data. So we've been really interested in getting at the heart of why this is true, and this pattern is also true in cognitive aging. Here we have data from Theodore and Castelletto's paper showing that sex is a variant in worsening memory and worsening processing speed, showing here that male sex is a risk factor for this decline. This has been shown true in other populations where, irrespective of ApoE genotype, male sex is associated with worsening memory across the adult lifespan. So we were interested in looking at what biological factors could influence this trend, and one of the factors could be the X chromosome. This is data from our lab, from Francesca, another grad student who recently showed
that in mice, adding an extra X chromosome leads to improved cognition in normal aging in mice. And this is true whether you have an X chromosome or a Y chromosome. So it's not the Y chromosome that's dragging things down, it's really the X chromosome that's boosting cognitive ability. So why would two X chromosomes matter in cognitive aging? In mammals that have two X chromosomes, one of them is randomly inactivated. However, there can be escape from X chromosome inactivation. This means that you're going to have biallelic expression or some additional expression of certain genes and certain cell types. So we were interested in two questions. First, does aging alter escape from XCI? And second, how do changes in escape contribute to female resilience in aging? And to do this, we used a really elegant mouse model that allowed us to track expression from the active and the inactive X chromosome. We did single nuclei RNA sequencing on the hippocampi of these mice in a young and old cohort, and we're able to stratify gene expression from both of the X's across different cell types. So first we asked the question, does aging preferentially change gene expression on the X chromosome? And we saw that the X chromosome uniquely had more differentially expressed genes compared to the autosomes. So this shows that the genes on the X chromosome are relevant to aging and the hippocampus as they're being differentially regulated. So while we're interested in sex differences, studying the X chromosome in itself is particularly captivating. Then we asked the question, does aging induce escape from X chromosome inactivation? And to start answering this question, we looked at expression from the inactive X chromosome. Here we focus in on the neurons and oligodendrocytes, as these were two different cell types in the hippocampus that had many differentially expressed genes. And we can see that while there's many changes on the XA, shown in black, there's also some changes from the XI. And the changes from the inactive X chromosome were particularly interesting as I highlighted genes like FTX, which is involved in XCI itself, but also genes like PLP1 and GPM6P, which are relevant to myelin. So the genes that are being biallelically or even just monoallelically increase or decrease from the XI in aging have important biological functions. So then we are interested in focusing more in escape by cell type and age. We used an escape model that focused on the XI expression over total X chromosome expression, and by doing this, we looked at all cell types and found a baseline group of escapees in the young population. However, a subset of these genes increase in their degree of escape with aging. There's a group that maintains, and then a very interesting group that's just activated from escapees with aging. And we focused in on these especially because they seemed like a really interesting group of genes being turned on from the XI in aging. So then we asked the question, does aging induce escape of any XI factors across all cell types? So here we see different genes that had changes in XI expression in the young compared to old, meaning that they have increased XI expression in the old in many different cell types as shown here. And we paid special attention to PLP1 as it had changes from the XI in many different cell types. Most robustly, we saw changes in oligodendrocytes, and we'll focus in on those a bit more later. And here we show XI expression changing in these cell types from young to old, and you can see that this upward trajectory holds true. So now, focusing in on PLP1, we asked the question, does increased XI PLP1 expression lead to a higher PLP1 dose in aged females? And first, looking at mice, when we look at expression from any X chromosomes in the mice, we see that old expression is increased only in XX mice. So this shows that there's a unique contribution of the XI. And additionally, what helps with many members of the Memory and Aging Center, with a special shout out to Rowan, we looked at human data in the parahippocampus region and saw that PLP1 expression was elevated in the female parahippocampus compared to males. So this gives some confidence that the
trends we're seeing in mice can be translated to humans. And this is really big because escape genes in mice aren't necessarily always escape genes in humans. So we were excited to see this. Then we asked the question, does increased PLP1 expression provide cognitive resilience in aging? And to do this, we use an AAV overexpression model that used an MVP promoter. So we were increasing the expression of PLP1 in oligodendrocytes, followed by a GFP marker. And here we show infection of this in the dentate gyrus, where the virus was injected, and overlap between OLIG2, which is a marker for oligodendrocytes, and the GFP virus in this region of the dentate gyrus. Overall, about 6% of the cells of the dentate gyrus had this co-infection. However, even just changing the 6% of cells had an impact on the mice behavior. Here we look at 2-trial Y-maze data, which is a way of looking at cognition. In old XY mice that were given this PLP1 overexpression, they showed improved cognition with the PLP1 overexpression. And this was also true in the XX mice. So this study shows that focusing on escape genes, while it may be a female phenomenon, can also have benefits for males and females alike. So to conclude, we saw that aging preferentially alters differentially expressed genes on the X chromosomes compared to the autosomes. Focused analysis of the XI showed changes in escape expression, including new escape with aging, including in myelin component PLP1. That PLP1 is increased in the aging parahippocampus of women, and that AAV-mediated elevation of PLP1 in oligodendrocytes of the dentate gyrus improved cognition in old mice of both sexes. So to conclude, the inactive X chromosome contributes to brain resilience and may inspire new therapeutics for men and women alike. And there's many different follow-up research questions we're hoping to pursue. First, what other age-activated escapees could provide cognitive resilience? If you think back a few slides, there were many other genes that had increased expression in aging, and it would be really interesting to focus in on these escapees in a similar manner as we did with PLP1. How similar are human and mouse escape patterns in aging? This will be a little more challenging to get at, but hopefully with some of the human blood and CSF found in the Memory and Aging Center, maybe we can start to get at the heart of this. And then finally, how variable is escape between individuals and aging? And as I've started to look at this final question in mice, it seems that some genes are really consistent between all of the mice, but some genes have a lot of variability. This variability may be present or absent in humans too. It'll be really interesting to look into it. So, want to thank everyone in the Duval Lab. Big thanks to Dina, and as well as all of our collaborations here.
Thank you for the talk, really amazing. I'm not familiar with brain stimulation research. Do you see that as kind of coming to clinic as a combination therapy? Do you think so?
Do I see this coming to the clinic as a combination therapy? Yeah, so brain stimulation doesn't necessarily work unless you engage the brain. I mean, there is some data that you can sort of passively apply it, but the type of stimulation that we're using with these electrical currents are very weak. We're talking around a milliamp. They're powered off watch batteries. So we're not driving neurons. We cannot actually cause a neuron to fire. So if you want to, and all these things are doing is biasing the way they fire. So if you're targeting a network, you need to kind of engage that network. So if anything, it's a crutch. We use this to facilitate performance on these tasks that we give them. So anytime someone is doing brain stimulation in our studies, we always have them engaging in the tasks so that they're trying to, so that the networks that are supposed to be used can work better, if that helps make sense. Now, there's other more powerful stimulation approaches, whether you're going invasively into the brain or even non-invasive, like magnetic stimulation or ultrasound. You can drive neurons, and that's a little bit different. But what we're looking here is a lot easier to implement and a lot safer, in my opinion.
I have a more technical question. How did you determine the dosing in your multitasking experiment? experimental paradigms versus your episodic memory, because it was like 15 minutes and there it was like almost an hour. So what was the plan for which made you decide?
Right. Yeah, so with the multitasking stuff, we base that off of previous literature and what we believe would be sufficient to engage the networks and cause change. Now, with the gamma stimulation, where we're targeting episodic memory, that was based off of, again, previous literature, but it was based off of sensory stimulation. So rather than using electrical, there's a whole other field, I mean, it's not massive, but there are multiple groups assessing the utility of flashing light or sound at these gamma frequencies. And they have some really interesting evidence in both, again, humans and animals that there may be some efficacy. And so we were basically just following suit with the idea that as a therapeutic, it would be really annoying to stare at a strobe light every day for an hour. So we thought, well, why don't we give them electrical stimulation, something that they could actually do while they're engaged in a task.
A short question, but like down the road, do you imagine like potential interventions for like healthy aging for women in like their like 30s or something, like starting early or?
Absolutely. I think the goal is, right, this is going to be true for aging research in general, but especially true here. There's not going to be some silver bullet. It's not like we're just going to have some single drug or something that you're going to give to every woman and it's just magically going to make their ovaries work properly for their whole lives. Rather, we're coming from a position of being not just like years behind or decades behind. We are generations behind understanding female bodies because we haven't been studying them. And so the goal is to understand how ovaries work at baseline, at different ages, meaning how they change with age, and that we can use that knowledge then to hopefully come up with a menu of options that would be interventions, that would be lifestyle changes, whatever, maybe a whole bunch of different things that would be appropriate for women to preserve or extend their ovarian function that would be different for someone who was 15 from someone who was 20 and had maybe like a history of PCOS, or someone like me who's 49 and about to enter menopause, but that we would understand in enough detail how ovaries work that we could just give women those options. So yes, and some of those definitely will be interventions and therapeutics. We're just not there yet because there's literally no IP. We haven't done the science to generate the IP to make the interventions. So, yeah.
Reported the mic, any questions?
Yeah, I didn't see other hands.
What do we know about the relationship between escape from the inactive X and menopause? You attributed some of the changes with age, but it may not be age, per se.
Yeah, it's hard to tell. The data we've looked at so far is mice or individuals who are much older than menopausal age, so I feel like we would need intermediates to do that. Some of our collaborators are focusing more on the hormonal aspect of these changes in aging, so I think we've started combining our genomic data sets and seeing what are some of the common genes or proteins that are changed, so I think that's the first step, because we really don't know.
Maybe I'll ask one last question and then maybe it's time to end. But I really loved, particularly in the last two sessions, we talk about sex, but actually there's an underlying biology that's a continuum there that is actually what we need to quantify when we're getting precise about what are the differences between males and females. And you each tapped on a component of sex differences, X chromosome, ovarian function. Can you maybe comment on how you think these might intersect or how we could think about them in a more holistic manner moving forward?
I think one of the really interesting tools to do this is that there's different mouse models that you can dissect, adding sex chromosomes, subtracting sex chromosomes, adding functions of the ovaries or testes, and really getting at what component is driving what, and then you can quantitatively see how much of a different variant is attributed to sex chromosomes or hormones, because people are on a continuum, so some of the data may be more or less relevant to them.
Beautiful. I guess I have two answers. One is when we're talking about aging ovaries, right? Suddenly the people in the audience who don't have ovaries sometimes tune out. Not always, but sometimes tune out. And I think it's important to point out that ovaries are actually, if you set aside sex differences, ovaries are like an amazing time machine. It's essentially what we need in the longevity space to test interventions. If you think about it, ovaries, at least all of the pathways that we know about that impact aging so far, are also operating in ovaries. It's just accelerated. So in a way, it's an accelerated model of human aging that manifests in women in their 20s and 30s. And so some of the work that we funded actually led to the first clinical study, the first ever clinical study in humans to look at ovarian endpoints, not for fertility, rather for ovarian aging. So that plants a flag and gives us the opportunity. It doesn't matter what the outcome of that study is. It means that we can then test interventions in this way in a relatively short period of time. So there's that. There's also this, there's so many associations that we don't understand in the little data that we have, and one is that age at menopause, age at natural menopause or reproductive span, correlates with overall lifespan. And this is of course on average, which means that on average, females who go through menopause later tend to live longer. And that extends to male brothers. So there's an underlying genetic component there that's important for both sexes. And so there's so much to be discovered here that's important not just for females, but also for males.
Rowan said I'm going to spend the next 5 minutes talking about vascular aging. Before I jump into the data, though, I want to get you as excited as I am, hopefully, about this concept of vascular aging. By showing you a couple of my favorite facts. So as of last year, we know now that nearly half of dementia cases can be attributed to modifiable risk. And specifically, these 14 factors listed here from the Lancet Commission. What's really inspiring to me in my work, and what's really pertinent to this talk today, is that 50% of these factors can be directly tied or linked to vascular health, really highlighting and pointing to vascular health as an important area of focus for promoting brain aging and for dementia prevention. But unfortunately, this is our current gold standard biomarker of cerebrovascular disease or vascular pathology. And these white matter hyperintensities here are markers of end-stage disease. And by this time, it's too late, and we need to be able to estimate vascular risk much earlier on. Moreover, there's increasing data that suggests that there are other pathologies that contribute to white matter hyperintensities. So we also need more precise estimates of vascular pathology and a more precise way to measure vascular aging. Another challenge in examining vascular health is that it's really complex and multifaceted. And while we typically think of it as a direct relationship from poor cardio to poor cerebrovascular health, it's likely that there's multiple pathways involved and multiple biological mechanisms underlying this connection, really further complicating this process. So as a lab, we think that fluid biomarkers are a really important step towards being able to tackle this complexity. And more specifically, they might allow us to more precisely estimate person-specific vascular risk. So I'm going to touch on a few projects that aim to leverage fluid biomarkers to better understand this cardiovascular to brain connection. So we'll look at an individual biomarker of cerebrovascular disease, as well as another study that looks at a more exploratory, multivariate approach and leverages large-scale proteomics to understand this relationship. So let's just jump into the first one. So today there are some biomarkers that exist for vascular pathology. PLGF in particular is a marker of endothelial dysfunction and has shown some promise as a biomarker of vascular-related cognitive impairment. More specifically, there's been a couple studies showing relationships between elevated PLGF and worse white matter outcomes, as well as increased functional impairment. But unfortunately, this data has been limited to cross-sectional analyses. So we wanted to look at this longitudinally in our functionally intact healthy aging cohort here at UCSF and examine whether the relationships between baseline plasma PLGF and cognitive trajectories, as well as white matter trajectories over time. And we found, surprisingly, that elevated PLGF did not relate or did not track with white matter metrics over time as expected. Instead, we found that there was a relationship between higher baseline PLGF and worse memory trajectories, as well as processing speed trajectories, leaving us with more questions about vascular aging. And what better way to dive into this deeper than to look at the whole proteome? So Rowan nicely teed this up for me today. This next project we're going to be talking about is a project in which we leverage blood proteomics to understand the heart-to-brain connection. And this was in the ROSMAP cohort, which is a clinical pathological cohort from Rush. And in short, what we did is we conducted a systematic review of the cardiovascular proteomics literature and identified 181 proteins that predicted cardiovascular risk. And then we looked at differential expression analyses, looking at the individual relationships between these cardiovascular risk proteins and cognitive and brain outcomes of interest. More specifically, our outcomes included cerebrovascular disease burden, global cognition, and systolic blood pressure, which was our proxy of cardiovascular risk. From there, we were able to identify several cardiovascular risk proteins that exhibited overlapping relationships between our outcomes of interest. But there was only one protein that associated with all of our three outcomes, SLAMF1. And so then we took this a step further, and we saw that there was a relationship with a random mediation model and found that cerebrovascular disease burden mediated 15% of the relationship between higher SLAMF1 and worse global cognition, suggesting that SLAMF1, which is an adaptive immune response protein, might represent a common biological link between heart and brain. So there were some other pathways popping up in some of our models, some of which Rowan's touched on today, cell homeostasis, immune response signaling, protein synthesis. But another thing that was popping up as we were running these models was how different things looked when we stratified by sex. So the proteins that were popping up looking at only males or only females were not the same. So we're excited to keep digging into these data and to look at person-specific factors that might be influencing these relationships. And I would love to chat with anybody who's interested in trying to understand the complexities of vascular aging with me. But for now, I'll just say thank you to our participants, families, and my amazing team here at the MAC. Thank you.
So I decided to do something a little bit different with my 5 minutes. I decided to focus more on my philosophy and values, and ultimately discuss why I feel hopeful about my line of inquiry. And why I think it's important to harness biology to overcome medical fatalism, and in doing so, reframe aging really as an opportunity for intervention. So as we know, it has been discussed, many medical conditions are really seen as an inevitable consequence of aging. And this is especially true for delirium, which is the focus of my research. And so delirium is often perceived as a sudden state of confusion that older adults are really expected to experience when they're hospitalized for illness and injury. And so some of you may know, I wrote about this observation when I was a medical trainee. And in this I wrote that delirium had an optics problem. What good was diagnosing delirium if you couldn't do anything about it? This attitude was a self-fulfilling prophecy. And this mindset really is an example of medical fatalism, this belief that nothing can really be done about it. And as a result, delirium has been overlooked in discussions about prevention and treatment. Very few neurologists even study delirium as a condition, even though it's a major risk factor for developing new or worsening dementia. And so what if we instead viewed aging as a biological process whose course could change? And so this is one of the main goals of geroscience, and this has been discussed. But what if delirium isn't just a warning sign of decline, this expected sign, but really an opportunity to better understand aging, and then could be viewed as a critical moment to change the course of health and recovery? And so I've led several prospective cohort studies that leverage this adaptive model of the metrics of aging, where traditionally much of the focus has been on describing phenotypic aging, which are the observable traits that we maybe think are associated with aging, and then quantifying also functional aging. So those are the ways that we see aging impact quality of life and other metrics of daily life and function. And so my goal has really been to add this biological aging step, getting at root mechanisms to delirium research. And so an example of phenotypic aging is delirium. And this is something that I've looked at in a few different cohorts, spanning older adults with hip fracture, internal medicine patients, and now looking at patients with traumatic brain injury, focusing more on neurological trauma as a neurologist, going back to neurological trauma. And root mechanisms, these are things that could be summarized through the canonical hallmarks of aging that we talk about in neuroscience. And in particular, I focused on measures of senescence and biological clocks, such as those that are measured with epigenetics and DNA methylation. And so we've measured these in blood and adipose tissue, and soon in collaborations with the Buck Institute, also in bone. And then we can look at measuring key clinical outcomes that matter to us and our patients, like cognitive and physical function. And so this past year, I helped lead an initiative through the NIH-funded Flynn Star program to describe one aging mechanism, which is inflammaging, or the chronic low-grade inflammation that's linked to advanced age. And we did this across 9 medical specialties. And what we did was create a transdisciplinary aging framework to demonstrate why it's important to break out of our subspecialty silos. And so I was a neurologist working with a dermatologist, working with rheumatology, working with gastroenterology, and all these different specialties. And so together we described cross-cutting putative pathways, proposed a transdisciplinary approach to research and clinical care, and how this collaboration could translate into more generalizable treatment strategies that could be tested in experiments, randomized controlled trials. And to do this successfully, I think it's also important that we talk about why diversity matters in this field. And I wanted to address language-related disparities in delirium, which this project is led by a very fabulous neurology R4, Dr. Mary Zanika, and leveraging our UCSF-wide delirium care pathway, where all non-ICU patients are screened for delirium each nursing shift. We look at about a year's worth of data divided by listed language preference, either English language preference or non-English language preference. And you can see here that in our initial cohort, overall the folks who were non-English language preference were on average older. And in models that adjusted for age, sex, and illness severity, we found that those with non-English language preference were more likely to have delirium on admission, have longer lengths of stay, and also higher rates of mortality during hospitalization. And so this will be discussed more in detail at an upcoming AAN plenary talk. And since one of the goals of this event is to foster collaboration, I also wanted to end with an open invitation to join me and Dr. Bradley Beauvais in our Sex and Gender Enriched, or SAGE, neurology program. And this is the first R25 research education program funded by NIPS to train neurologists how to advance neurological care and research informed by sex and gender. And so this grant enables us to host interdisciplinary workshops on topics related to sex and gender, like one I hosted late last year that was focused on traumatic brain injury in women across the lifespan and the intersection with delirium and dementia. And we're always looking for more topics. So please reach out to me if you're interested in participating in any of these collaborative opportunities.
So my name is Claire. I'm a postdoctoral fellow here at the MAC, and I'm really excited to be here today to talk about some preliminary data from our study that aims to look at the optimal parameters of physical activity sessions for brain health in older adults. And so there's lots of research out there to suggest that higher total levels of physical activity associate with better cognition and brain outcomes in aging. But one of the most common questions we get asked is, how much is enough? How intensely do I have to go to achieve those outcomes? And it turns out that we still don't have very good answers on this, something that has likely contributed to the mixed success of physical activity intervention trials. And so part of the argument is that maybe we need to further refine the type of physical activity being prescribed in those trials, and understand person-specific differences that might contribute for better success. And so the overarching aim of this project is to use objective step count data and machine learning to examine the optimal parameters of physical activity sessions that associate with brain health outcomes. And to start with, we've just asked, within overall activity level, how important are structured physical activity sessions? And among people who engage in those sessions, what is the relative importance of specific session parameters like duration and frequency? And so we have 329 older adults who all have 30 days of minute level actigraphy data and a range of cognitive and brain imaging outcomes. And I'm just going to briefly give you an idea of how we derived the sessions from this minute step count data and the parameters that go into these models. And so this is an example of an hour of minute level step count data. And we defined a physical activity session as at least 10 minutes where step count was above 40 steps per minute, which is considered a threshold for purposeful low intensity physical activity. Once that criteria was met, we also allowed for short breaks within physical activity sessions, because in the real world, people stop to take a breather or they order a coffee while they're out on their walk. And so now we've defined this physical activity session, and everything else outside of that session is considered out of session activity. And for those sessions, we could define their duration, their cadence, their time of day, their session, total session steps, as well as their frequency within a week. And so firstly, we just asked, do people who engaged in sessions during that 30 days differ on any brain health outcomes compared to those who don't engage in any sessions? And what we found is that those who engaged in physical activity sessions had lower total white matter hyperintensity burden than those who didn't. And this makes a lot of sense. White matter health is closely tied to cardiovascular health, something that physical activity is a known contributor to. We didn't see any strong associations across our analysis with the other volumetric or cognitive outcomes in this unimpaired sample, and so I'm just going to focus on white matter hyperintensities for the remainder. So then we asked, at a broad level, how important is in-session activity versus out-of-session activity? And so just to show here first that in general, most people do most of their steps out of sessions compared to in sessions. And we used random forest machine learning with nested cross-validation to assess the relative importance of these two predictors in the model. And what we found is that in-session steps were more important for predicting white matter hyperintensity burden than out-of-session steps. And so just for the purpose of visualization here, I've plotted the relationships linearly, where you can see a steeper negative slope against white matter hyperintensity burden for in-session steps compared to out-of-session steps. So next we asked for people who engaged in sessions, what was the relative importance of specific exercise parameters? So we substituted the in-session steps predictor in the model for all of the specific session parameters that make it up. And what we found is that session frequency was the most important, followed by duration and then cadence, with out-of-session steps continuing to be the least important predictor. And so again, for visualization, I've just plotted the top two predictors there linearly, where it looks like higher frequency and longer session durations are associated with lower white matter hyperintensity burden. So some preliminary conclusions: engagement in structured physical activity sessions is associated with white matter health in older adults. And despite most of people's steps happening outside of sessions, in-session steps were the most important for predicting white matter hyperintensity burden. And finally, that session frequency might be the most important parameter to optimize for physical activity interventions. In terms of our next steps for this project, there are some other activity parameters that we'd like to include, including how consistent a physical activity routine is throughout a week. As mentioned, we'd also like to look at person-specific factors that might influence the optimal parameters, and most importantly, we'd like to work towards determining parameter cutoffs to inform physical activity recommendations. And just very briefly, thank you to everyone involved in this project, particularly Emily and Caitlin, and all the research coordinators who work extremely hard to collect all of this data.
Thank you very much for the opportunity to speak here. And today we've heard a bit about sex differences and just now about exercise. And I'm talking about sex-specific molecular mechanisms in exercise using a mouse model to look at the aged mouse hippocampus. And so as we've talked about, aging leads to cognitive decline, but this is in a sex-specific manner. It is thought that women are more cognitively resilient as they age. But conversely, the prevalence of dementia is twice-fold in females versus males, indicating this sexual dimorphism with aging and with brain aging. So our lab studies the hippocampus, the region responsible for learning and memory. We use the mouse model here. And the hippocampus is made of the trisynaptic circuit of excitatory neurons that help with encoding learning and memory. And with aging, we see these gross cellular changes with the loss of neuronal connectivity or changes in regenerative capacity. But we know that we can leverage some lifestyle interventions to investigate cognitive rejuvenation in the hippocampus. So this could, for this, we use mice, a mouse model where we give them a running wheel and they will voluntarily exercise. And we can see that in this mouse model, we can rejuvenate the aged brain and lead to increased cognitive function. To then determine which of these gene expression changes are perhaps malleable or modifiable, we wondered what are the molecular changes that are underlying age-related cognitive decline in rejuvenation. And we wondered, given the sex-specific differences in aging, and that 50% of the population is female, what do these molecular changes look like between males and females? So our work is, one of our projects is looking at how sex impacts molecular or exercise-induced molecular rejuvenation in the aging hippocampus. So for this, we started to functionally test how exercise impacts cognitive decline. So we used two cohorts of mice here, males and females, looking at young and aged, and then aged exercise mice where they undergo this voluntary exercise paradigm for 6 weeks. We then tested them using a number of cognitive behavioral tasks, and we found that exercise was able to rejuvenate the aged brain and lead to improved cognitive function in both the male and female mice. And then we utilized new RNA sequencing technologies to look at these molecular changes. So we isolated these hippocampi, and then we sorted out nuclei and used single nucleus RNA sequencing and used a 10x genomics platform. And so for this is a UMAP projection looking at the number of the nuclei that we were able to isolate from the hippocampi of male and female mice. And we were able to identify a number of these different cell types, including the neurons of the trisynaptic circuit. And in males, we see that with aging, there's this large decrease in a number of genes, but that a number of genes are increased with exercise. And when we look at the overlap of differential gene expression in aging versus exercise, we see around 2,000 genes here that are changing. When we look at that directionality, we see that a number of those genes that are changing in one direction with age are changing in the other direction with exercise. So for example, if it's going down with age, it's now going up with exercise. And a lot of these genes are in the dentate gyrus neurons of the hippocampus. And when we then look at what those genes related to, we see that there's a lot of changes in synapse plasticity or RNA splicing. And excitingly, that exercise seems to rejuvenate a number of these pathways. So here, just looking at a heat map of some of these genes that are within these GO terms, you can see this partial rejuvenation to a more youthful state of gene expression. And with females, we also see a number of changes with aging and exercise, particularly more so now in oligodendrocytes, as well as the dentate gyrus neurons. And we look at the overlap of these genes, we also see this bidirectional regulation. So these genes that are changing with aging that are changing the opposite direction with exercise. And we focus in on oligodendrocytes for our female data set, given that there's this difference in the cell type that's really modulated by sex. And we see there's a lot of changes in synapse plasticity, so oligodendrocytes are supporting these neurons with these gene expression changes, and that is changing with age and exercise. We also see that exercise is able to partially rejuvenate the aging transcriptional changes in these female oligodendrocytes as well. So changes to synapse organization seem to be rejuvenated, as well as synaptic translation. And so then we wanted to know what are the similarities and differences between male and female exercise-induced rejuvenation. So if we look at those genes that changed, that were changed in both aging and exercise in males or females, we actually see there's only a really small number of those genes that are changed in both males and females, only 87 of these genes, so really small overlap. And when we look at what those genes relate to, it no longer relates to synapses, like we've shown before. It's regulation of membrane potential or phosphatase activity, things that aren't relating to these synapses. So while synapses seem to be changing in both males and females, it seems to be working through completely different genetic or transcriptional mechanisms. So much more analysis and work is to be done here. But with that, I would like to thank my lab, thank you to Valvita, my mentor, and to Greg and Juliana, people in my lab that have worked with this project. Greg is here and he's an exercise expert. And thank you to collaborations and his listening.
My name is Joanna. I'm a fourth year neurosciences student. I'm in Dr. Judy Pa's lab. She really wishes she could be here today, but the San Diego fires were really close, so unfortunately she couldn't. But today I'm presenting on circadian rhythms, aerobic exercise, and Alzheimer's. So just to provide a very quick background, as we've discussed, there's several modifiable lifestyle risk factors for Alzheimer's, ones that we could target in order to decrease one's individual risk to develop the disease. I look at specifically circadian rhythm dysfunction and physical inactivity, and I want to know how they're involved with Alzheimer's pathology, amyloid beta and tau, as well as the ApoE4 risk gene. And I want to understand these relationships prior to the onset of irreversible brain atrophy and cognitive decline, so that we might be able to identify circadian-based therapies to intervene. To understand these relationships, I've used the LearnIt trial, which was a 6-month aerobic exercise intervention trial led by my lab back when Judy was at USC. The sample is 68 sedentary older adults with objective early cognitive impairment, and participants are randomized either into the exercise intervention or the health education compared to a placebo that remains sedentary. At baseline and the 6-month follow-up, participants wear accelerometers for about 1 month. They undergo A beta and tau PET, tests of cognition, and E4 phenotyping. So first, with the baseline data, I wanted to determine how are circadian rhythms related to our biomarkers and cognition. And very quickly, the measures to note here is intradaily variability is a measure of rhythm fragmentation, and after time is a timing measure where a higher number means a later time. As for our outcome measures, we have A beta PET as a composite, tau PET over Braak staging regions of interest 1, 2, and 3, 4, and then we use verbal memory as our measure of cognition. So shown here are the added variable plots to visualize the regression models ran between our circadian rhythm measures and our pathological outcomes. In A, B, and C, we see that after time was negatively associated with all of our pathological outcomes, such that an earlier peak activity time is associated with higher pathological burden. And then in D, we see that higher intradaily variability, our fragmentation measure, was associated with higher tau. We wanted to look at the moderating effects of our covariates in these relationships. So we see in A that ApoE4 carrier status modified the association between after time and A beta burden, such that the E4 carriers in yellow have a steeper slope. We also see that age and sex modify the association between intradaily variability and tau burden, such that this relationship increases with age and disproportionately affects men compared to women. As for our cognitive measure, similar to the pathology relationships, we see that an earlier after time was associated with the worst performance in verbal memory. And in this relationship, we wanted to look at some potential mediators, and I was able to identify that tau in Braak regions of interest 1, 2, shown on the left, and 3, 4, shown on the right, mediated the association between after time and short delay verbal memory. This shows that tau could be a potential mediator in this relationship, or a mechanism in this relationship. Now, very quickly, preliminary findings that I just submitted for AIC, so I don't have too much on this, put it together on Friday, but I wanted to determine how long-term moderate aerobic exercise over 6 months impacts circadian rhythms. Oops. The measures to note here are 2 of circadian strength, called amplitude and Macer, and another one of circadian timing, called M10 start time. For those 3 measures, I calculated percent change between baseline and 6 months, and then analysis of covariance showed a significant positive difference in percent change for all 3 variables, such that the 6-month aerobic intervention compared to the placebo control strengthened circadian rhythms, shown in A and B with the higher amplitude and Macer in the exercise group, and it also shifted rhythms later compared to the placebo control, shown by the higher M10 start time. So what do we get from this takeaways involving precision-based medicine is that 1, older adults with earlier and more fragmented rhythms may be at higher risk for developing Alzheimer's pathological hallmarks, amyloid beta, tau, and cognition, or having reduced cognition, and this may be occurring through a tau mechanism. The individuals who are at higher risk may be older individuals, males, and ApoE4 carriers. And then our really new exciting results is that long-term aerobic exercise both strengthens and alters the timing of circadian rhythms, which shows that exercise could be a promising point of intervention to entrain circadian rhythms in order to improve Alzheimer's pathological and cognitive outcomes. And with that, here's the PAW Lab. Thank you to everyone in it, my funding, and also to UCSF for bringing me out here today.
That was really fun. I have lots of questions individually, but maybe thinking across these projects, there was a really strong, person-specific, especially sex differences theme, and maybe you can all comment about what are some commonalities of the biology maybe that's underlying some of the sex differences you're seeing in exercise or circadian differences or vascular health. and where there's some divergences, and so how could that inform, our next steps in this space? Again, sex being just a binary variable that we use, and you all are getting deep on the biology.
I can start. We have a higher percentage of women with Alzheimer's, but it's really interesting that across sleep and circadian rhythm findings, we typically see that men have it worse. It's kind of one of those few things where I think men do have it worse in terms of Alzheimer's related outcomes. So that's always been very interesting to me, and I've thought about it being just sex specific things that women experience that may be influencing that. I'm not too sure exactly, but I want to look into it more. But it has been kind of interesting to think about that in comparison to all the other things that men experience that would lead to higher, sorry, I lost my train of thought, but that's all I have for now.
I feel like I'm kind of biased because I love cardiovascular health. I think it ties a lot of these things together. And when we think about age, like some of the things that we talked about, age, sex, cardiovascular health, physical activity, and sleep, I think they're all tied together still by cardiovascular health. And we know, with increasing age, there's more cardiovascular risk. There's, sex differences in cardiovascular risk, such that at younger ages, you see males have higher risk, and then there's more data showing that there's a flip in postmenopausal women. So I know we talked about this a lot, and I definitely think there's something to that, but in terms of like the systems, biological mechanisms at play, I don't know, I think the immune response pathway we've talked about a lot today popped up in Rowan's presentation, and it was one of our hits as well. So something to think about, but yeah, it's interesting.
Yeah, I don't have too much to add on top of that, I guess, but given, all of these differences that, both presenters have already spoken about, I think we're really excited to look at these person-specific differences when we're looking at these physical activity recommendations in our project. So, given different risk profiles and biological mechanisms at play, I think we might see different patterns and different recommendations coming through.
Yeah, it's all very interesting. I just wanted to add with the mouse model that we're going to start, kind of functionally testing some of these genes, and we've been looking at the different ones, but also looking at what's similar between males and females, and one of these master regulators, and if that's different between the sexes or not.
Thank you so much for amazing talks. My question is related to exercise. I think it's very exciting to see these very promising results. But I always think about, a lot of my work is international, and thinking about sort of the role of intentionality of exercise has always been something on my mind. Like, for example, if you think about construction workers, right? They do a lot of exercise, what we would consider physical exercise, or somebody who works night shifts all the time, right? Their circadian rhythms are likely disrupted. So I was wondering if you have any thoughts about the role of occupation and overall the social exposome. Would you think that these effects are sustained? Would you think that there's some sort of a differential pathway? Any thoughts are welcome. Thank you.
So just understanding your question, it was around how so occupations might affect circadian rhythms and how exercise might play into, physical activity might play into that.
Whether you think the effects, positive effects of exercise would be sustained when exercise is not necessarily intentional, when it's part of your occupation.
Sure, yeah, that's a really good question. I think, so part of our project's aim is to try to capture physical activity in all of its forms, so not just intentional physical activity that you set out to do in a session, but also physical activity that you might do as part of a job. I think, in terms of what we found so far, we had this very crude measure of whether you're doing physical activity in the morning or in the afternoon, versus, other research presented today, which had a very more fine-grained measure of when activity was happening in the day, which I think is really important. We didn't see strong effects of, time of day of exercise on brain health outcomes, but I know, other research presented here has shown effects, so maybe you want to speak to that further.
Sorry, can you say that?
So just in terms of the circadian kind of differences that you saw in your project that were different to the we didn't see it as much of an important factor in health.
Yeah. Sorry, maybe just repeat the last part of the question.
Yes,
I kind of got all jumbled up.
I'm curious what your thoughts are about sort of not whether somebody follows, let's say, an unnatural circadian rhythm. Do you think that might impact your results? You know, when somebody has to work night shifts and it's not necessarily sort of their natural way of engaging in exercise, whether aerobic or not, whether sustained or not.
Yeah, absolutely. Well, we see all across the board that people who have disrupted circadian rhythms, whether it's night shifts or just irregular schedules, we have a lot of higher pathology. It leads to different diseases. And so I would say that, yeah, of course, if we have disruptions between people who do night shifts, you're going to see big differences there. And then in terms of exercise, I am very curious about the timing of exercise. That's something I'm planning on looking into in the longitudinal data coming up, is what about the timing of exercise? What about the people working out in the morning versus the middle of the day versus later? So that's what I plan to do next, is kind of separate it into different times and see how that affects people's rhythms and pathological and cognitive outcomes. So I'm curious to find out, but I just got access to longitudinal data like 2 weeks ago to throw it together for AIC, so it's very preliminary.
Hi, really great talks. I love that the theme here is around, both modifiable risk factors and these person-specific factors based on which you might make more specific recommendations for modifying these risk factors. But I'm curious to hear all of your thoughts on, the benefits of making these very person-specific recommendations that might be harder to deliver because it requires some level of baseline assessment in some way, whether it's their baseline behaviors, their genetics, demographics, and other things, versus these sort of blanket recommendations that are maybe more, easier to deliver to large swaths of the population worldwide. So just curious to hear your thoughts on that.
Or I can just do a quick start on that, is that precision interventions for individual outcomes is something I'm really interested in. Some of the people in we're doing a virtual reality health study in the Paul Lab right now. People have come to me and asked, can you kind of like walk me through my sleep profile, my circadian rhythm profile, how much am I waking up in the middle of the night? And I think that's kind of a fun ability to be able to talk to the person and see what they want improved and what they need to work on. But I would say, that requires a one-on-one intervention, which is kind of what I want to do as a career someday. But also I think that's valuable, but so is these bigger blanket statements like this population might be more at risk compared to this population. If you're an E4 carrier, these relationships could be stronger and you should be thinking about your circadian rhythms and your sleep. So I would say like both are really valuable to have in this space.
Yeah, I think it would be amiss not to mention, I think, health disparities in the context of this, conversation, because sometimes these blanket statements are not possible for people, and I think it's important to think of it like both ways. We need to get person-specific, but we also need to be thinking about access and what people are capable of doing and the different types of exercises that are even possible for people. I think it was a good point to bring up how different things are even between, different countries. And but I think even within, males and females, like starting there, that's so broad, that's 50-50, and we're learning that things are so different between males and females. And I think sometimes getting more granular is really important, and I think that there's a lot of potential, therapeutic benefits from that, more tailored recommendations. But I think we've been so broad in general that now is the time to get more specific within age groups, within sexes and ethnicities and so on. So it's exciting times.
Well, this isn't work that I discussed today, but past work has involved the delirium care pathway and more its actual implementation at UCSF and sort of hospital-wide. And I think that this provides an interesting model because there are so many risk factors for delirium. This is something that we've published on in a systematic review. There's at least 30 existing risk factors, and then in terms of acute precipitants, you can think about at least 100 or so acute precipitants for delirium. And so these pathways really do need to be more broadly distributed and really try to get at some of the more common risk factors for delirium. But then you can think about that person's individual, more specific risk factors to get that more precision and potentially have a greater impact. So I sort of see it as you can, with these kinds of pathways, you can prevent 30% or so of delirium cases, and 30% is a long way from 100. And so you can have a widely applicable pathway that'll get you partly there, but really to get all the way there, we need to be more precise in terms of how we're rolling it out.
Yeah, I think it was said that both blanket statements and looking at precision medicine are both important. In some of the mouse work that I'm familiar with, it's just only been male cohorts of mice for a really long time. So a lot of these like exercise-induced factors or other youthful factors that have been identified and tested, they were usually done in male cohorts. So I think it's, and then perhaps when it gets to humans and then there's like, why doesn't it work? And it's because those, preclinical things were only in a certain subset. So I think both are really important, and it could be that things that we are identifying are applicable to many people, and it could also be that it's really specific. So important to look at both.